This video describes how to do ICM Autofit for X-ray electron density maps. The ICM X-Ray Autofit method is an automated way to fit a ligand into electron density. The tool combines the powerful ICM docking algorithm with an electron density fitting algorithm. The ICM X-Ray Autofit method includes the following features: a soft docking energy function. Traditional rigid docking methods may struggle when the binding pocket exhibits some flexibility. The soft docking energy function accounts for minor conformational changes, allowing for a better fit within the binding pocket and also avoiding artificially high strain on the ligand. There's also an intra and inter-ligand interaction energy function. The method considers both the internal strain of the ligand and how it interacts with surrounding residues in the binding site. This ensures that the final pose not only fits the density, but is also chemically reasonable and energetically favorable. There's weighted electron density contributions, so not all parts of the electron density contribute equally to the ligand placement. The method applies a weighting scheme to prioritize high confidence regions of the density, improving the accuracy of the fit. And then there's an electron density filtering to exclude protein receptor atoms. So to prevent interference from the protein structure itself, the electron density used for ligand fitting is filtered. The step removes areas occupied by receptor atoms, ensuring that only relevant density is considered for ligand placement. So let's see how to do this practically in ICM. So we are going to read in a PDB file, 1xBB with a ligand. We're going to read in the electron density maps for this. So you go to Tools, X-Ray, get electron density map for 1xBB. So the maps are now here. We can contour that map. So if you just select the ligand and then contour around that. So you select the ligand, go to Tools, X-Ray, contour electron density. You can change different sigma levels and the coloring of those sigma levels and go OK. So if we zoom in, you see in this example there's some electron density around this. This is the sort of solvent exposed part of the kinase. There's a couple of sites where maybe the electron density could fit better. Some of these sites are for waters. We display the waters. But others are not filled. So we can see if we can improve this fit a little bit. So we first need to convert the PDB into an ICM object. Right click, convert PDB, replace the original, go OK. Next, we need to move the ligand out of the pocket. Move from object, and I'm just going to rename it for clarity. We select the ligand. We go to the docking menu, new project. Give the project a name. The receptor object is 1xBB. And we're going to define site around the ligand. So now we have the green crosses around the ligand. And just go OK. This is the probe where the simulation will start. And the next thing is the purple box. But we won't change any of those for this example. So it's calculating the maps. So the next step is to tell ICM we want to dock not only to the crystal structure maps, but also to the electron density in the pocket. So we go to the docking menu and you choose X-ray density here. And just go, you find the map. The map is at 1xBB and go OK. If you want to check whether it's actually using the density or not, you can go to your docking project files. So here, the docking project directory, and you can see we have an extra map called XR for the X-ray. And you can also just check in the .dtp file, which is the control table file with all the different parameters. If you just search for density weight here, and if it's set to 1, then you know that it's using the electron density as well. So now it's ready to dock. So we can just extract the ligand, 2D drawing, go to docking, dock chemical table, just go OK. When the docking is finished, you'll see a note saying that the docking is finished. Click OK, and it generates the hit list. And single click on the hit list to load the ligand in. So the docked one is colored by, I'm going to color C only, magenta, and the yellow one is the X-ray structure. So you see it tried in this sort of solvent exposed region. The crystal structure has the ligand pointing out into the solvent, which is fine. Could be the correct orientation, but there is a little bit of density here, which may indicate that this part of the ligand maybe makes this orientation. So it's using not only the electron density, but also the maps for the docking of the receptor as well. Thank you.